Hello, everyone. It's lovely that we're having so many people from around the world joining us today. I am Associate Professor Joanne Passmore from the University of Cape Town in South Africa. It's 4 p.m. in the afternoon here in Africa, Central African time. So it's a real privilege of mine to introduce our speaker today, Dr. Yasmine Belkaid from the NIH in the USA. She is a distinguished investigator at the NIH, and she is going to be talking to us today about the interaction or the mechanism of the interaction between the microbiome and host immunity. Her lab explores fundamental mechanisms that regulate tissue homeostasis and host immune responses. And we are really privileged to have her presenting today. At the end of the session, we are going to have a Q&A, and I just wanted to urge the attendees to please use the Q&A tab at the bottom of the Zoom call, because we'll be focusing questions from the Q&A section, or raise your hand and ask questions. And I just urge people to introduce who they are at the beginning of asking their questions. That would be great. And then I just wanted to invite all attendees today to take note of the IUIS conference being held next year, 2023, in Cape Town, South Africa, in our summer. Shout out from Cape Town as a local Cape Townian. We're really welcoming people to register, to submit abstracts. Registration isn't open yet, but please watch the website. And I think at the end of the talks, there will be a post up with the details. So with that, I just wanted to open the talk up for Dr. Belkaid, and we really look forward to your presentation today. So thank you very much.
Thank you very much, and I'm going to just start to share my slides. Can you basically unshare your slide for me to be able to do that? Wonderful. Share, and I'm going to go to. So first, it's my great pleasure to be here today, and I'm very happy to be part of this wonderful seminar series, and actually very much looking forward to be at the meeting next year in Cape Town. So I'm just very much like Joanne mentioned, I'm going to discuss with you some of the work in the laboratory trying to understand the interplay between the immune system and the microbiota, and in particular to try to tackle the really challenging questions of mechanism of interaction. And we have to all agree that although we have now a great understanding of this alliance between the microbiota and immune system, it's going to take us as a community an enormous amount of years to be able to really figure out how these microbes are interacting specifically with tissue in the context of many biological settings. And the point that I wanted to highlight first is actually a very short summary of what has now become a really well-established phenomenon, and it's our understanding that the immune system and the microbiota are profoundly entwined. And to have an immune system that is actually perfectly fit to control infection, develop response to vaccine, or even, as it was recently shown, respond to immune checkpoint therapy, the microbiota is actually clearly an ally of these immune responses. The microbiota can actually allow the development of the immune system, it allows the fine-tuning of the tissues, and it allows also the development of B cells and T cell response to pathogens. So really some of the most spectacular link that has been highlighted recently is in the context of immune checkpoint therapy. And some recent work that was done at the NIH in part, but also in other places, has highlighted that in the context of humans that are responding to immune checkpoint therapy in the context of melanoma, the microbiota can actually be taken from these responders and transferred to non-responders and reconstitute responsiveness, which is actually a remarkable proof of principle of the role of the microbiota as an adjuvant in the context of human responses. But of course, as many of us do know, this relationship can be threatened. And in many circumstances, aberrant reactivity to the microbiota has been linked to inflammatory disorders. The best example of that would be, of course, inflammatory bowel disease, but it's of course clear now that actually this is actually a companion of numerous inflammatory settings. And if the microbiota is not always the cause of inflammation, it is at the minimum the amplifier of inflammatory process because of the sheer number of innate ligands and antigen present in those communities of microbes. But the question I'm going to discuss with you today is not about just this link between the microbiota and immune system, but is more a fundamental question that started to really perplex us in the laboratory over the last few years. So beautiful work that had been done by January and Polly Matzinger and many others since then has helped us to understand the framework of how we develop an immune responses. And what had been postulated, and is actually very much true for the most part, is that you do need inflammation and tissue damage for the immune system to realize that there is an invading agent and you need to unleash immune responses. And this class of immunity is, of course, necessary for the control of pathogen invasion. It is also the very same class of immunity that can be dysregulated and lead to immunopathology in inflammatory disease or in the context of aberrant response to self. But this class of immunity really didn't fit with what we began to understand about the microbiota. The microbiota colonize all barrier surfaces early in life, and you constantly acquire new microbes as adults. However, these microbes are not causing any form of inflammation, but they are clearly able to induce T cells and B cell responses. And this was a paradox. You know, how does these microbes that are not invaders are able to induce immunity? So the question we decided to explore in the laboratory are twofold. And the first one is to try to understand what are these strange property and function of an immune responses that is not here to control an invading agent. There is no invading agent. And the second one, what will be the mechanism by which the microbiota engage in such a quiescent way with the immune system. So just to summarize the work of quite a few investigators in my own laboratory that had the privilege to work with really incredible scientists over the last few years. So first, Shruti Naik, who now is actually leading a very successful program at NYU. When she came in my laboratory as a graduate student, was intrigued by the link between the microbiota and the immune system of the skin. And she decided to utilize this tissue for mechanistic insight, making the postulate that this tissue has a lower biomass of microbe, and it may be actually simpler to infer causality in the context of this simpler structure in contrasting to the GI tract. So what Shruti was able to show is that if you apply a new microbe at the surface of the skin, and we have now confirmed that for any new microbe that you apply at the surface of the skin, you can induce an immune responses that could be CD8, CD4, gamma delta, mate cells, and in some cases, all of them. These T cells are able to accumulate within the tissue and be long-term resident cells. They can communicate with the epithelium via cytokine production, allowing the tissue to produce more antimicrobial peptide, thereby allowing the immune system to be more responding to subsequent infection. So what Shruti was able to show is that an adaptive response to the microbiota, a cognate response to microbiota-derived antigen, can actually promote broadly innate responses of the tissue. What was subsequently shown by Oli and Mike and John is that this class of immunity was really different from an immunity to pathogen. These T cells are highly plastic and can change their function locally based on the stimuli. And what they were able to show is that the very same cells that are able to promote antimicrobial defenses could also be rewired and promote epithelial repair in the context of injury. So what was actually postulated by this investigator in the lab is that immunity to the microbiota was non-inflammatory, but nonetheless was highly efficient and was able to broadly promote tissue response and physiology. But this, of course, led us with numerous questions, and in particular, the mechanism by which those microbiota are able to engage the immune system. And the first hint of the mechanism came from the work in the laboratory of Samira Tamutunur when she was looking at how the tissue responded to an association with the microbe. And what she was able to show in a very surprising way is that although you didn't have inflammation, what you had is a very strong antiviral signature. It's almost like the tissue was infected by the virus. And it was quite perplexing to us. Perplexing, but not really surprising. Work that had been done by other investigators in the field, including Akiko Iwasaki and David Artis, highlighted that the gut microbiota was able to broadly highlight and enhance antiviral responses in the lung, for example. However, the mechanism of this link between the microbiota and antiviral signature had remained unclear. And this led us to revisit a really fundamental concept of biology, is that, of course, we are composed of exogenous microbiota, that will be the bacteria, the fungi, the viruses, and the protozoans. What we also have is an endogenous virome. 10% of human genome is actually composed of remnant of retroviral infection that have embedded themselves in our genome, around 8% in the mice system. These elements, of course, have been subjected to enormous amount of control to prevent their reactivation, fragmentation, control, epigenetic control of these elements. However, some of those have maintained translational activity and have been linked to various biological functions. So this led us to postulate that there may be a link between the exogenous microbiota and the endogenous retroviruses. And this question in laboratory was addressed by a very talented group of fellows, Jalma and Sid. Sid being a bioinformatician and Jalma a bench scientist. They paired up to tackle this complex question. But they first started with a very simple experiment in which they took a microbe, Staph epi in this case, but this was done with other microbes too, applied the surface of the skin of an animal that was not a germ-free animal. Those animals contain their own microbiota, and they mimic the acquisition of a new microbe. And what they were able to show is if you purify keratinocyte a few days later, is that you have a significant, although discrete, upregulation of ERVs within the keratinocytes. So, of course, the next question is to try to see if this ERV had anything to do with the response to the microbiota. And as we know, these elements can have a life cycle, and they could be potentially converted to RNA, but they can also, if there is an active reverse transcriptase linked to this element, be converted into double-stranded DNA that could be putatively sensed as an innate ligand in the cytoplasm. So to address this question, Jalma and Sid utilize a treatment that many of the people in this audience will be familiar with, that will be in the context of HIV prevention and treatment, which is an anti-reverse transcriptase inhibitor. And this will actually block the ability of those reverse transcriptase to convert those elements into DNA, and therefore DNA to be sensed by the innate immune system. So when you apply Staph epi at the surface of the skin, you have an accumulation of T cells within the skin compartment, and you can see then just blocking the anti-RT in this animal prevented the acquisition of this neo-response to the microbiota. This can be visualized in a more clear way by imaging, where you can see the accumulation of CD4 and CD8 T cells to Staph epi in the context of association, and how this anti-RT treatment profoundly inhibit the ability of those T cells to accumulate within the tissue compartment. So quite surprisingly, response to a new microbe is coupled with reverse transcriptase activity of the host, and thereby likely expression of those ERVs. So we all know that from the seminal work done by numerous investigators, then when the DNA accumulates in the cytoplasm, it can be sensed as a sign of viral infection and can be sensed by the canonical SIGA-STING pathway, leading to a set of responses programming antiviral responses. So to make a long story short, we confirm using full knockout, then the SIGAS and STING pathway were required, but our next question was to try to understand which cells were responsible for the sensing of these ERVs in the context of commensal association. And to make a long story short, we utilized numerous CRE systems, and were able to show that keratinocyte expression of STING was absolutely required for these responses. And this can be summarized here. If you remove STING expression only on keratinocytes, in the K14 CRE mice, you can see that the basal activation and accumulation of CD8 T cells is already impaired in animals without this pathway, supporting the idea that the tonic accumulation of those tissue-resident memory cells is under this control. But you can see that the response to Staph epi is also profoundly inhibited in the context of Staph epi association. The same thing is true for made cells, a population that we have shown to be highly induced by Staph epi, and a population of cells that is highly represented in human and highly represented in the skin of mice. You can see that in this context, there is a decrease of the tonic accumulation of the cells and the profound inhibition of these responses for Staph epi. Similar responses observed in gamma delta T cells that are entirely depending on this pathway. So this demonstrates really then the keratinocyte is actually required for the accumulation of the commensal specific T cells. Our next question, of course, was to try to understand how these responses to ERV could also explain the ability of the microbiota to promote inflammatory response in some context. And for doing that, for addressing this question, we utilize a very simple approach that is linked to a change in dietary component of the host. And I'm going to explain to you what I'm meaning here. As we have shown now extensively in the laboratory, if you apply a new microbe at the surface of the skin, there's absolutely no inflammation, which is quite reassuring for many of us that are constantly acquiring new microbes. But we had actually shown a few years ago, and I revisited that with Staph epi, if you change the diet of the host and you actually led the animal to eat high-fat diet, only for a few weeks, that's really preceding metabolic syndrome, just for 3 to 6 weeks. These completely transform the ability of the host to sense the microbiota away from these homeostatic responses to an inflammatory. And I'm showing that here. You apply the same microbe at the surface of the skin, and you can see now then there's this massive inflammation that occurs in response to association of the microbe. So you can see here the thickening of the ear, and in particular the thickening of the keratinocyte layer and proliferation of these cells in response to Staph epi only when you have a high diet. So quite remarkable to think about. Changing the diet can be sufficient to convert this homeostatic relationship of the host with the microbiota from homeostatic to inflammatory. Using this approach, Jalma and Sid decided to look at the ERV expression by keratinocyte, and they were able to show, and especially looking at the different part of those viruses, the gag, the propole, or the envelope, then if you apply Staph epi at the surface of the skin of an animal that is fed a high-fat diet, there is really a massive upregulation of ERVs that can actually be visualized here. This can be also further visualized by using another form of representation when you can see on the right the massive upregulation of ERVs in the context of Staph epi with high-fat diet as opposed to non-high-fat diet. Underlined, you will see actually elements that are predicted to have an active reverse transcriptase. And this was done in collaboration with the leader in the field, George Cassiotes, that help to predict those elements and the ERV expression of these elements. So moving forward, we decided to block once again the reverse transcriptase activity to see if this could at least in part control this inflammatory response to the microbiota. So in red is inflammatory response developed by the host in response to the microbiota and high-fat diet. And you can see the significant downregulation of this inflammatory response by the RT treatment, the anti-RT treatment. It is not an absolute shutdown, but nonetheless, there's a significant contribution of reverse transcriptase activity to the inflammatory process seen in this setting. So once again, we found that this reverse transcriptase activity and activity of the microbiota is highly entwined with ERV activity of the host. So quite surprisingly, we found that the microbiota and the ERV really in this extraordinary multi-kingdom dialogue is able to control immunity in the tissues. And somehow it's quite remarkable to think that the microbiota found a way to co-opt those ancient viral partners as a way to engage with the immune system of the host and to develop this homeostatic response that we know is so beneficial for tissue responses. We found that ERV expression is actually required for response to the skin microbiota, and the cGASTing pathway can promote homeostatic response to the microbiota. So I used to refer as the microbiota as the adjuvant of the immune system, but I think this needs to be rephrased. The real adjuvant of the immune system are the endogenous retroviruses that are really present in the host. So the model we have at the moment is the following one. We find that actually the microbiota is able to communicate with keratinocytes, and we have actually now evidence that this occurs, and this is not the point of this discussion today, but via specific ligand at the surface of the microbiota that can interact specifically with TLR2. Downstream of TLR2, to mechanisms that remain to be fully understood, there is an upregulation, a very discrete upregulation of ERVs that lead to the accumulation of DNA in the cytoplasm via the reverse transcript activity and the sensing by the cGASTing pathway, leading to the activation of keratinocyte and the engagement of the immune responses. What is quite remarkable about this form of immunity is clearly this happens in a very discrete way in which the cells are engaged, but not at a high level enough to lead to inflammation. And this really allows the development of these homeostatic responses. This is change in the context of alteration of the host, and we have here exemplified that in the context of a dietary intervention. When the keratinocytes sense lipid at a high level, and we are now trying to understand really the mechanism, this explains this phenomenon, the response to the microbiota now leads to a synergy of signal and major aberrant expression of ERVs. And in this context, the ERVs are no longer homeostatic, but now promoting an inflammatory responses via the cGASTing pathway, leading to pathology. So the model we have at the moment is that really the level of expression of ERV in the tissues is actually very much the one that dictates the ability of the immune system to sense the microbiota in an homeostatic as opposed to inflammatory way. And we're now exploring this question in numerous settings. First, we're trying to understand this extraordinary link with the diet and ERV, and I'm going to show you some of the examples of how we're going forward. We're trying to understand also how in individuals that are genetically predisposed to have a defect in expression and antiviral responses, ERV can actually potentially be dysregulated, leading to inflammation. And also understanding the crosstalk between infection and ERV expression in the skin. And really to highlight the point that we're actually moving forward, we have now evidence that this phenomenon is not restricted to the skin. In fact, we find that in the lung in particular, this response, if anything, is even more dependent on VRP expression by epithelial cells. And we have early evidence, and this is also the case within the context of the vaginal epithelium and also in the context of the gut. So we are now developing numerous projects in the laboratory to tackle the role of ERV expression in various barrier surfaces and how this actually dictates the ability of the host to develop immunity to pathogen, to the microbiota, and how this regulation of these responses can lead to inflammatory responses. But before moving forward, I just wanted to highlight very shortly some work that is done in the laboratory by Verena Lick and Kevin Hall, in which we're beginning to address this question in the context of human setting. And this actually is done in the context of a really extraordinary cohort of individuals that we're lucky enough to work with at the NIH. These are actually healthy individuals that are brought to the clinical center, and they will be placed either into a vegan diet or a ketogenic diet. And what is remarkable about this study, it is probably the most highly controlled nutritional intervention that ever been done, because the individuals are in the clinical center, only eat what is provided to them. Let's face it, everybody cheats with diet, so it's actually a very good way to control what people eat. And also the same individuals are really looked via the lens of two different diets. And we were lucky enough to be able to collect a large number of data sets and information on this individual. The first part of this study has been recently published by the group of Kevin, highlighting the metabolic alteration in this host. And we are now exploring and developing a manuscript on looking at the immune impact of these diets. But the reason I'm actually discussing that is twofold. First, I just wanted to highlight very briefly the remarkable observation that Verena has done, is that quite surprisingly, when you have the same individual on a vegan and a ketogenic, they have a paradoxical responses, with vegan being able to push dramatically innate responses, and the ketogenic is more able to push adaptive responses. So it's quite striking to think that something as simple than 2 weeks dietary intervention can shape the immune system in such a profound and conserved way among all the participants. But the reason I'm highlighting that today is because although it has been shown before and we have confirmed that ERV are highly individual specific, what we're also able to show at the level of certain cells is that the element that are the most changed in the context of dietary intervention only after 2 weeks are actually ERVs. So quite remarkably, we found that there is this extraordinary link between ERV and nutrition. To which extent this explains how nutrition is actually affecting the immune system remains to be addressed, of course, but nonetheless, knowing the potential of this element in regulating the immune system, this is a really tempting hypothesis to think that actually somehow there is a privileged interaction between ERV expression and nutrition, and that could at least in part explain the ability of nutrition to actually engage the immune system. Another point that is remarkable and need to be explored is how this heterogeneity in human could also explain the differential ability of the individual to respond to various infection, vaccine responses, or innate responses. So we are now in the process of actually trying to develop and further this exploration at the level of different cells in the host and in the level of tissue, and try to understand how we could potentially make sense of the extraordinary heterogeneity immune landscape of human via the lens of this ERV expression. But before finishing, I just wanted to spend the next few minutes really highlighting a very emerging story in the laboratory that is really the understanding of a new role of the microbiota in the immune system. And as I mentioned before, the microbiota is able to tap into these extraordinary resources that is the adaptive immune system to control host physiology. And we have actually shown that this actually controls barrier function, antimicrobial defenses, epithelial repair. We wanted to actually keep digging and try to see if you could uncover a novel function for immunity to the microbiota. And this question in laboratory was done by Michel Enamorados, who became fascinated by the interaction between Staphylococcus aureus and the immune system. And the reason he was so interested is because we all think about Staph aureus as a pathogen, but the reality is Staph aureus is extremely diverse. In fact, we are all likely, a vast majority of us, colonized by this microbe in a very stable way. And a lot of those strains will not cause infection. And even in the one that can cause infection, they may be coexisting with the host for years before, in some circumstances, causing infection. So Michel postulated that this microbe that can, for the most part, coexist with us as a member of the microbiota may be well suited to induce an immune responses that can prepare the host for subsequent tissue damage, including tissue damage caused by the very same infection. So to address this question, Michel developed first a tool to track immune response to Staph aureus. And in this case, he transferred these transgenic T cells in which all the T cells are specific for the Staph aureus antigen into an animal that contained polyclonal responses, and using actually labeling system was able to track these cells in tissue. So the cells, after you had applied the microbe at the surface of the skin, accumulate within the skin, showing once again that there is an adaptive response to the Staph aureus. Not surprisingly, the vast majority of these cells adopt this type 17 behavior, which means the cells are able to produce cytokines that are very good at controlling the epithelium. And this is not surprising because many microbes that colonize us tend to push those type 17 response. But what was surprising is when Michel started to look at the behavior of the cells in the tissue. And he did that by using mice that were engineered to visualize various systems. And in this specific case, to make a long story short, I'm showing you mice in which all the nerves that are CGRP expressors, which are the sensory nerves in the periphery, have been engineered to actually be visualized by microscopy. So these nerves are here visualized in purple, and the commensal specific T cells are visualized in yellow. So you transfer the T cells, and you can see in the context of Staph aureus association that there was this intriguing association between the T cells specific for the microbiota and the sensory neurons. And you can visualize this kind of association in some of those cells. This can be visualized on the right when you can see that these cells that are commensal specific are in closer proximity from sensory neuron than they will be from the endothelium. So this, pointed maybe, although it could have been just correlative, to a role of the commensal specific with sensory neurons. So Michel sorted the commensal specific T cells and looked at the gene expression of these commensal specific T cells and was able to show that these T cells were intriguingly wired. First, they express a tissue repair and wound healing, not surprising, but what they do also express is the nerve interaction and regeneration signature, pointing to the possibility that these cells may be involved in neuronal regeneration. So to address this question, Michel utilized a very simple approach, that is to look at a punch biopsy in the ear of the animal and allow us to visualize the regeneration of sensory neuron. And those sensory neurons and the neurons in general will be visualized as this white in the periphery, creating this ring that is really surrounding the edge of the wound, slowly closing the injury. If you have any doubt that the immune system is important tissue repair, just look at this slide when you can see the massive infiltrate of CD4 T cells, but also of regulatory T cells that really surround the edge of the wound. So using this approach, Michel decided to see if having Staph aureus somehow, strangely enough, could accelerate the repair of the nerves. The first experiment is quite simple. You take an animal control that doesn't have Staph aureus or apply Staph aureus a few weeks before the injury. The microbe persists in a very low biomass at this stage, and 10 days post-injury, really look at the sensory neuron regeneration. You can see that there is an increased accumulation of CD4 T cells if you have Staph aureus, but importantly, you can see that there is a densification of the fibers that are linked to the regeneration of the neurons if you have Staph aureus association. And this can be quantified on the right. So having Staph aureus at the surface of the skin is linked to accelerated neuronal regeneration. But of course, this could be linked to anything. It could be linked to the Staph aureus itself, producing molecules that favor regeneration. So to try to see if the T cells specific for the microbiota could be important for these responses, Michel developed a strategy in which he deleted the ability of T cells entering in the tissue to produce this type 17 cytokine I have mentioned before as so important for the function of commensal specific T cells using an OX4 decree. And using this approach, we find that the T cells can delete this program only when they enter the tissue. So control mice develop, of course, a regeneration that actually can be visualized here. If you apply Staph aureus at the surface of the skin, you can see the massive densification of the nerves, showing that there is an acceleration of the repair. But if you delete the ability of those T cells to produce type 17 cytokine, you can see that this regeneration is profoundly inhibited. And this can be quantified on the right. Having commensal specific T cells induce the repair. Having a defect in the ability of these cells to produce cytokine locally is linked to a defect in the ability of the cells to respond. So this really pointed to CD4 T cells being responsible for the ability of Staphylococcus to promote tissue repair. But of course, sensory neurons are wired in a way where all the information and all the mechanism linked to their function happen really upstream at the level of the dorsal root ganglia. So to try to see if somehow these nerves can respond directly to type 17, Michel did another experiment in which he visualized the expression of genes by RNA scope at the level of the ganglia themselves. And these ganglia are very important, the DRG, because not only they're actually connecting the skin or other peripheral tissue to the spinal cord, but this is also important for the conveying of this information to the central nervous system. So previous work had actually shown that there is one transcription factor that is very important for the regeneration of neuron, and is ATF3. And if you do an injury at the level of the skin, you can see an upregulation of these factors after 3 days. And this is something that Michel confirmed in the setting we have actually utilized, and this is actually his own data, confirming the published paper. What was really, really interesting, and I don't know what happened here, there is actually a break on the, sorry, doesn't matter. For some reason I moved something. So anyway, so what is really interesting here is you can see that these DRG that are injured expressing ATF3 also express the IL-17 receptor A, which means then they acquire the ability to sense and respond directly to the very same cytokine that is produced by commensal specific T cells. And this can be visualized here when you can see that there is a coordinated response of the IL-17 receptor A with the ATF3 expression. We also confirmed that this was not only happening in this specific model of injury, but happened also in other models of neuronal injury. So it's appeared to be a very conserved response to sensory neuron injury. So the next question is, of course, to try to see if IL-17 could directly promote the sensory neuron regeneration. Because, of course, when we think about repair, we very much often think about macrophages that have been shown to be very important for the regeneration of tissues. We rarely think about T cells able to produce a factor that can act directly on the injured organ. So to address this question, what Michel did is to utilize an animal in which you could remove the receptor for IL-17 only on the sensory neuron. He applied an animal that can sense IL-17 or cannot sense IL-17 with Staphylococcus. You can see the acceleration of the repair visualized by the beta tubulin, that is a pan-neuronal marker, but also CGRP staining really that shows this is actually happening at the level of the sensory neuron. But if you remove the ability of sensory neuron to sense IL-17 produced by the T cells, you have now a defect in repair that can be also confirmed at the level of the sensory neuron. This is actually seen here when you can see a significant decrease in the regeneration of the neurons if you do not have the ability to sense IL-17, and also the sensory neurons as visualized by CGRP expression. So the model that we have is the following one. We constantly acquiring new microbes, including Staphylococcus, but many others. This microbe can induce cognate responses that we found to have the ability to accumulate at the proximity of sensory neuron. It's quite remarkable, and this is not actually only happening with Staphylococcus. We have found that other microbes, such as Staph epi and many others, are really, really tightly bound to those sensory neurons, and we're now exploring the mechanism of interaction of this interaction. What we also find is in the context of an injury, the microbes are still there. Remember that they are commensals. So they will actually, the antigen will actually be dragged in the context of the tissue, allowing the local recall of these antigen-specific responses that can produce a large amount of IL-17. At the same time, you have IL-17 production. The DRG upregulate IL-17 receptor mRNA that lead to IL-17 receptor expression, allowing the sensory neuron to now sense this IL-17 and co-opt it to promote this regeneration of neurons. And this really leads to the entire regeneration of the sensory neuron networks that is of prime importance for numerous reasons. First, recovery of sensation is an extremely important phenomenon. But more than that, the sensory neuron coordinate action of the immune system, and they're also important for the regeneration of the vasculature and the regeneration of the epithelium. So to really regenerate the tissue as a totality, you need to actually regenerate the sensory neuron that will seed the path of all the structure to be regenerated. So to conclude my talk, I just wanted to highlight the fact that I showed a few things that both intrigued us and surprised us. The first one is that, strangely enough, to be able to communicate with the immune system, the microbiota need to utilize an ancient viral partner that are the ERVs, and to utilize that as a way to actually engage its adaptive responses. So it's a remarkable association that we are quite fascinated in laboratory and now exploring in numerous tissues in the context of human settings. The microbiota is able to induce these cognate responses that have the ability to expand and actually develop and accumulate in the tissue. So somehow it can capitalize on the power of the adaptive immune responses to control tissue physiology, the ability of the cells to actually in plastic, to expand the context of exposure to antigen. And this leads to numerous functions: enhance immune defenses via the enhance of antimicrobial peptide, but also enhance epithelial repair. And as I have shown here to the work of Michel, enhance neuronal regeneration. So many questions remain to be addressed, of course. And one of them that emerged from this work is that IL-17 can be surprisingly maybe a target for the regeneration of sensory neurons, something that is, of course, of critical need in patients for the sensory recovery, a phenomenon that happened in the context of injury, but also in the context of surgery. What happened also in the context of diabetic wound when actually the wound is not healing. And finally, what happened in the context of neuropathy when people have aberrant sensing? Could it be also caused by an aberrant regeneration of the sensory neuron that is linked to an excess of IL-17? So this is actually a line of investigation we are now further exploring. But more generally, what I wanted to highlight here is the remarkable ability of the microbiota to develop an immunity that has the ability to just promote tissue physiology, but also to induce its multi-system repair, which is something that I think has not actually been uncovered before and seems to be a unique feature of this extraordinary immunity to the microbiota. So to conclude my talk, I will say that working on the microbiota made me a perpetual student and make my laboratory a perpetual students of this phenomenon. We're really looking at them, we're exploring. We are not smart enough to figure out how the system works, but the microbe have done that. And by really looking at how does microbe coexist with us and manipulate the immune system, we may be able to uncover novel therapeutic approaches and to try to begin to understand the true logic of the immune system of tissues by really looking at how this microbe have actually tapped into this evolutionary relationship to develop with extraordinary physiological immune responses. So I think there is years ahead of us in mechanistic discovery. I think we have been maybe a bit too impatient in the field and thinking that this was over. The field is actually growing and the field is actually going in mechanistic insight. And I think there is an enormous potential of discovery over the next few years. It's a very humbling field, but it's, I think, an extraordinary potential to really potentially develop strategies that are aiming at restoring physiology by taking into account this really ecologic relationship with our microbes. So on that, I have to finish my talk by thanking the extraordinary people that have been privileged enough to work with over the last few years. I have worked with some of the most extraordinary trainees and students I can imagine, and I'm extremely grateful for the work they are doing and they have been doing. Many of them have started their lab across the planet and are doing very, very well, and I'm very proud of them. For the purpose of this talk, I wanted to highlight the work of Jalma Lima and Sid, who have both done the work on the RV, and Michel, who has done the work on the sensory neuron, a remarkable group of fellows, very soon applying for independent positions. So if any of you here are actually looking for extraordinary fellows to start their lab, these people are really unique. Wonderful collaboration, Isaac Xu, and Claire Lepichon and Alex Chesner at the NIH, all of them have actually allowed us to really tap into this extraordinary relationship with the nervous system, with the immune system. Jos Cassiote is an expert in ERV and has been really our major lead in the project. And Michael Fischbach and Erin Chen, long-term collaborators on the mechanism of interaction of the microbiota with the immune system. And on that, I have to thank you all and we'll be happy to take any questions.
Wow, I'm blown away. That was such an excellent talk and it gave us so much to think about. And I think that's reflected by there's more than 10 very, very insightful questions in the Q&A. I would like to invite people who have posted their questions to pose those questions to Yasmine, and maybe to start with Francesca Ronchi. Apologies if I'm pronouncing your name wrong. So the question was, do you think that the effect that Staph aureus has on the skin in the context of inducing Th17 is important in neuronal regeneration, is specific for Staph aureus on the skin, or other commensals could do that? The example she's given is Staph epididymis.
Yeah. So I don't really think at all this is specific from Staph aureus. Remember, something that I have mentioned is actually the microbiota tend to promote those IL-17 responses very broadly. I think that any microbe that induce type 17, which means most of them, may be really capable to promote the physiological production of this cytokine that is so important for this reason. And I think that's where the framework of thinking about the microbiota differ from thinking about pathogen. None of us have evolved to live with one microbe. We have lived to work with this extraordinary redundancy. All of us have different microbes. It will be a dangerous proposition for the host to actually depend on one microbe. And I think we had this very naive approach over the last few years for all of us to try to find the microbe causing whatever. We have in thousands of species. All of us have different. So this microbe converge to common pathway that can really, promote tissue physiology. And we have actually now evidence that this is likely the case from Staph epi, and I bet any microbe that induce 17 can do the same thing. We zoom on those microbes because we have developed antigen-specific tools. And I feel like to be able to go deeper, we needed to also assess and prove specificity. But I do think that any microbe inducing this response will have likely the same phenomenon. And we're now working with bioengineering to just deliver IL-17 formulated in a way that allows us to target the neurons and see if we can do that directly by bypassing the microbe and just using 17 slowly deliver at the site of injury.
Very, very interesting. So there's a question in the Q&A from Samuel Alvarez Argudas. Apologies, you're going to have to forgive me. That was an awesome presentation. They're really curious about the involvement of DRGs in the repair mechanism, as was I. Do you think that this DRG activation could be related in some way with pathogen dissemination transmission? Do you think that this mechanism could also be present in the gut, lung, and other epithelial surfaces?
Yeah, it's a very clever question because, of course, we do know that those sensory neurons can actually be sites of chronic viral infection. And that's a very intriguing question. And we haven't done the experiment, but that could be very, very interesting to see if you have a persistent chronic viral infection and you have a damage of the nerves. Does this somehow link to reactivation? Something that is a very interesting question in the context we know, for example, then there is actually reactivation of zona in French, which is, of course, I forgot the name of it, but anyway, so there is reactivation of viruses in human that can happen in those chronic inflammatory responses. So I think it would be interesting to test. We haven't done that, but I think it's a very clever idea. And to try to see if somehow those good phenomena that happen in the context of regeneration can, in some individual, have the negative consequences to induce reactivation of virus. Very, very interesting question. Now, does it happen also in other tissue? We haven't tested. We're now looking at spinal cord injury and looking at those more severe form of neuronal damage to see by using 17 if we can actually now promote regeneration. But I think there is no reason to believe that this phenomenon may be restricted to the skin. My long experience working in various tissue is canonical fundamental mechanisms tend to be conserved. And there is, of course, some specificity in the cells that come to the things, but at the end of the day, I cannot believe that there would be a unique mechanism of repairs to the skin, especially when we think about sensory neuron. I don't even believe that there is only one mechanism, and I do believe that there may be numerous paths that could lead to regeneration. So I think it may be very interesting to test lung injury and to look at regeneration or gut something to explore.
So just from my perspective, what really intrigued me, and I work in the reproductive tract, so it's sort of another mucosal surface, but I was intrigued by, the nerve mucosa microbe intersection, and I'm a cytokine person, so I know something like VEGF, which is a vascularization cytokine, how you looking at causality between the interplay between vascularization in repair versus the nerve brain axes. I don't know if I'm posing that very clearly.
No, you're asking the question the right way, and that's exactly the complexity of working in tissue. There's no linearity. There is actually original event, but then there are loops and self-amplifying loops. And I think we're all struggling in how we do actually really develop rigorous science to address not only what we used to do in the past, which is one cytokine, one phenomenon, how do we actually really trying to understand the complexity? And I'm not telling you have the answer, but we're doing by one, and we're trying slowly to integrate more and more. So going to the vaginal environment or the female reproductive tract, incredible environment because incredibly high in ERV expression. And we know, in fact, something that is quite remarkable, and I don't know how many people know that, but the development of the placenta was actually the acquisition of retroelements. So we became mammal, really, because of endogenous retroviruses, which I think blows my mind. But also these tissues are really entwined with a lot of ERV activities. And we have recent work in the laboratory, when we utilize an anti-RT treatment, we actually shut down a lot of those homeostatic response in the vaginal compartment, but also in the uterus. So it's very early for us to see how much this, in addition, links to the sensory nervous system. But my point is, everything is always linked. And I think that's what we need to actually begin to address, is go to the mess and trying to develop rigorous approach to tackle the mess, because we all have actually been limited by the framework of a field, but a tissue and a host is all this system working and constantly with each other. And when you think about regeneration, in particular, this is a very multi-system function. So I don't have any answer, but my bet is there will be extraordinary link between the sensory neuron of the vaginal environment and the repair physiology and the commensal microbiota and so forth. So I think much more remain to be explored by young investigators in this audience that will actually not only develop mechanistic insight, but develop modeling and being able to actually have strong computational approaches to tackle those questions. But this is where the field is going. The field is going to integration. And I think we're now ready to go for it.
Absolutely. And also just the impressive tools we've got at our fingertips at the moment.
I know. I think so much needs to be done. And I wanted to say if there's any trainees here, when I hear sometimes people say, oh, the field is too competitive, there's nothing, I'm like, are you kidding? There's so many open questions.
Absolutely.
There's so many new things that needs to be discovered. So it's competitive if we all work on the same thing. But let's face it, we know very little about almost everything. So how about going to those directions? And I tell you from experience, there is a lot of space.
Yeah, absolutely. So very early on, Rao Divi asked a couple of questions about whether the microbiota could contribute to tolerance to pathogens in the way the body develops tolerance to self.
So it is, I can actually talk about tolerance to itself. So for example, the microbiota, and it has been shown, if you have acquired the microbiota early in life, so we all have those seeding microbes that we have. Some of them will develop responses, some of them we end up tolerizing. And it's not really clear to know what dictate what. We all know that the birth is actually linked to accumulation of microbes from the mother, and then of course at the weaning time, the child will develop a neo response to those microbes. So clearly not tolerization there, but nonetheless there is some tolerization to some of these microbes. So that remains an open question. How do you actually make this dichotomy? And it's so it depends probably of when you see the microbe and how you see the microbe. Now, are the microbiota promoting tolerance to pathogen? It's difficult to know, but one thing that is certain is the microbiota promote repair, promote barrier function, and thereby may prevent entrance of some of those pathogens. So it may not be a classical tolerance, but it may be ability to actually shut down the system before it's actually entering and damaging. I don't really think there is a clear evidence that the microbiota directly promote tolerance to the pathogen. I don't really believe that tolerance to microbe is always a great idea. That's actually, it may happen in the context of long chronic infection, and we have revealed that in the context of Leishmania and others. But for the most part, I think you may want to maintain the system alert, including to the microbiota, because the very same microbe that live in your skin can become a source of sepsis. And I think for us, if we were to develop dramatic tolerization or elimination or repertoire of those antigens that are required, we will probably be all dead from sepsis by now. So I think I think the best function of the microbiota is to reinforce the barrier, the shield, and taking advantage of the ability to have strong structures, but also making sure that immunity to this microbe is tamed and is actually very homeostatic. I could be proven wrong. That's what the yeah.
So there was a question which I've sort of cherry picking opportunistically here, but there was one question about the acute or chronic use of antibiotics and how this would affect the system, and whether you've got thoughts or data about that.
I think it's been now very well shown by numerous investigators, and including us, but we are not actually leading this kind of project. One thing that I want to highlight, and I think I want to, because I'm in an infectious disease environment and I'm working with infectious disease doctor, let's all remember, antibiotics are necessary to save lives. So this is not, I do not want to actually send the wrong message that the reason we're still alive at the density we are and women do not die in childbirth is probably because we have antibiotics. So let's just make sure that we give that said, antibiotics should not be misused, should not be overused. And there is absolute evidence that random utilization of antibiotics or chronic utilization of antibiotics for no reasons can have long-term dysbiotic effect. So, and we know that this actually can be linked to metabolic dysregulation, especially early in life, can lead to predisposition to inflammatory responses, very much because we know that having this alliance with the microbe is required. The antibiotics have other effects. Let's remember another thing. Every cells we have has a mitochondria. And what is a mitochondria? Mitochondria is of bacterial origin. A lot of antibiotics actually affect mitochondrial function. So let's all remember, we should never actually just have a dogmatic vision on anything. Antibiotics are still a required tool. That said, development of the next generation of antibiotics that will be more targeted is well needed. And more importantly, how do we actually compensate or repair for the damage that could be transiently caused by antibiotic? What kind of actually repair system, what kind of nutritional impact, what kind of fuel we need to give to the right microbiota is where we need to move in medicine. And I think that's really something that, unfortunately, in my opinion, is not moving fast enough.
Yeah, noted. Related to that, from Milton Maciel, there's a natural experiment going on in the summation with subjects taking Truvada, the antiretroviral drug. Yep. And do you have access to human samples to explore the same mechanism in humans?
Yes, we're going to have that. So I've been starting to discuss with Areni Sereti, that is a wonderful clinician at the NIH that works, that has access to patient. And we actually going to do something that hasn't been really done before, strangely enough, is really to have a longitudinal study in which we really have access to tissue prior to treatment and after the treatment. We do know that actually those retroviral treatments can actually have, they are incredibly important and have been actually life-saving, but they also have consequences. And we do not know if some of it is actually linked somehow to some of the effect on the immune system or others. And I think also it's important to understand what is the difference between exposure to these drugs early in life as opposed to actually in adult stage. Maybe in adult stage, you have generated enough and commensal specific T cells to no longer need this relationship. And maybe this is actually something that may be more of importance early on. Regardless, we're going to be able now to follow and have access to tissue before and after. In mice model, we have actually now shown that if you utilize this drug in mice, you actually decrease the quality of the immune responses, both type 1 and type 17, in every compartment we have looked in: the vaginal compartment, the lung, the skin, and the GI tract. So, of course, the mice is a very different system than the human, but nonetheless, I think it could be very fascinating to actually do a thorough analysis of what happened in the immune system.
So there was a question, sorry, I'm trying to pull it up quickly, because you mentioned leprosy, but somebody in the chat said, have you thought of exploring leprosy infection as a model for peripheral neuropathy? There's evidence that the skin microbiome differences in patients with different form of leprosy versus controls.
I think it would be fascinating. I mean, leprosy is linked to this profound degeneration of the tissue and eventually and actually also, I do not think that there is pain, but I know that there is actually shutdown of the systems. And of course, collapse of the tissue, which involved then the neurons that are also damaged. So we have not thought about it. Of course, leprosy will have to go to, I forgot what is the animal model for that, which actually could be done. But of course, we could have access to patient and maybe some biopsy. But coming back to this point, is actually 17 or slow infusion of 17 something that may allow the regeneration and facilitate regeneration in some of those things. Another system I'm thinking about is diabetic wound, where you have this failure of repair. Could reinfusing some of this wood with some of those molecules and other molecules allowing regeneration, I think would be fascinating. So we haven't considered, I think it's armadillo. I don't know. There's a very strange animal that you utilize for leprosy, and I'm embarrassed to say I don't really remember, if I say something stupid, but I do know that it doesn't infect those small rodents for the mechanistic side. But I think having access, armadillo. I just got correct, I was right. But I think if someone actually is interested to explore those questions, has access to patient or has access to armadillo, we're more than happy to actually collaborate.
Okay, great. Hand up to Steve for his persistence, but he really wants to know the answer to this question. Do you know if the same mechanism is at play in hidradenitis suppurativa, where IL-17 is known to be involved?
I have to just here with my joker, because I have no idea what this pathology is, and I have to admit that humbly that I'm not a clinician. So I cannot actually say anything. What I can say, maybe turning the question around, is we're now doing some work on psoriasis, which is not what you're just saying. And we find that, psoriasis is linked to this pain and actually these massive type 17 responses. And experimentally, we have evidence now, and early evidence, that this aberrant type 17 is also linked to this massive regeneration of the neurons that infiltrates the epithelium and may cause those neuropathy. So I don't know if the link of this disease. I'm more than happy to follow up offline if you have any question, or if you can send me information about the disease. But my point is, coming to before, everything is going to be always contributing. It may not be to the mechanism I'm discussing, but how could we not think that the neurons are going to be involved with every single phenomenon? How could not think that actually the very same microbiota that is colonizing every tissue will not be? How could not think that the tissue resident T cells will not be? So to my opinion, I can say yes to everything, not knowing what, not knowing how, but I will say yes. And I think that's the message I want to send to the students. Let's look at the connection. Let's not actually create barrier in how we explore questions. Common inflammatory disease where etiology is unknown, but 17 blocker effective. Then it will be very interesting to look at it. Thanks, Steve.
We definitely learned an enormous amount from both your talk, but also from some of the questions.
I know.
It's 3 minutes before the hour, and I am aware of our hard deadline. Have you got any closing thoughts that you wanted to share? Otherwise, I'm going to hand over to the organizers.
No, I just going to say very quickly something that I said before. I think this is a field that is infinite, that is a field that has space for people from every single specialty, and it's a field that allows people to collaborate across field. So I'm encouraging people to really, even if they don't work on the microbiota, to at least never deny the importance of this within the context of whatever disease or inflammatory setting they're working in. We are at the stage where we're having tools to explore questions in a more and more in-depth way, both in human and experimental model. So my advice is, it is an open field with an enormous, enormous potential for a new career to develop or career to reinvent themselves. So that's my little final.
So on behalf of the organizers of the seminars, I am so pleased to have listened to your talk. I learned an enormous amount. I'm inspired, even though I'm not a young academic. I just wanted to give one last shout out to the International Union for Immunology Societies taking place in Cape Town. I'm hoping, Yasmine, that you'll attend as well, and I'm just urging everyone to keep an eye on the website and look for when the registration opens later this year. But this was such an important topic, and I really thoroughly enjoyed the multiple directions you took to tackle this important interaction.
And I just wanted to say, if I haven't been able to answer your question, feel free to reach out to me by email. My email is on my website. It is not a secret email. So reach out and I'm more than happy to continue the conversation. And it was a pleasure to be here. And I'm really looking forward to be in Cape Town in a year.
Brilliant. And I'm ordering good weather and a lovely venue. And I really look forward to seeing everyone next year in November.
Bye-bye.
Bye, everyone.